Okay, so now we are going to talk about gene transfer, and we are going to first kind of broadly speak. In this chapter, we're going to be talking about horizontal gene transfer. And so this is the process of transferring DNA between bacteria that are not really related to each other. So, this can either be between bacteria that are different species. So you can have an E. coli bacteria transfer DNA to a Caulobacter bacteria. Or if they're somewhat related, so you can have one type of Bacillus subtilis transfer DNA to a different species of Bacillus tuberculosis, and the idea, though, is that you transfer DNA between members of the species or different species or closely related species, but or even within the same species, but they're not necessarily related to each other. And so that's kind of what the horizontal gene transfer means. So this is distinguished between vertical gene transfer, which is typically transferred by inheritance, where you have cell division and you have like a mother-daughter relationship. But horizontal gene transfer, so when you think about gene transfer in humans, that's the primary way that you have gene transfer. But in bacteria, the primary way is horizontal, or not. It's very important. Horizontal is very important to evolution of bacteria. This is very important for things like antibiotic resistance, for example. And there's three main ways that DNA is exchanged, and they are called transformation, conjugation, and transduction. And so these are mediated by different ways. So one is external DNA that is taken up by a cell. One is through the use of plasmids and pili in order to transfer the plasmids. And then the other is viral particles can actually transfer DNA from one cell to another. So we are first going to cover conjugation. And there's actually two different ways of doing conjugation that we are going to cover. So in order to understand how conjugation works, you have to understand the F factor. So this is the first example of a plasmid that conferred fertility that was studied in the lab. So the F factor allows a bacterial cell to become kind of sexually active, where it's able to now transfer DNA from one cell to another. And so this is unidirectional. So the bacteria can only transfer DNA in one direction using the F factor. And so we distinguish this between F plus and F minus cells. So F plus cells are the donors. They're the ones that have the F factor plasmid, and they're actually able to transfer their DNA to another cell. F minus cells are the recipients, so they receive the DNA, and then their cell is typically genetically modified. So again, the F plus cell has the fertility factor, and this actually confers the ability to create pili, which allows it to donate DNA and conjugate to other cells. And so this is kind of what we're showing here. So there's this plasmid that has the F factor, and then this F factor has genes that are able to create the pili, and so this bridge actually allows DNA to transfer from one cell to another. So E. coli may or may not contain the F factor, so some E. coli have this F factor plasmid, some do not. So if the F factor is present, then the cell forms these sex pilus, and this is how it's able to donate genetic DNA from one cell to the other. But importantly, F factor is also transferred from the F plus cell to the F minus recipient. And so what this does is it converts the recipient to the F plus state. So it becomes viral, where you're able to spread the F factor. And so this is part of the reason why the F factor can spread so readily, is because every time it conjugates, this F minus cell is converted to an F plus cell, and then the cell is able to also undergo conjugation. So I think how this works is just most visually, is best to understand just kind of like visually. So you have two different cells. So one of these cells is an F plus cell, and one of these cells is an F minus cell. So this F plus cell is able to create the pili, and it creates an actual bridge in between the two bacterial cells. So this conjugation has occurred. So you replicate this F factor, so this F factor undergoes DNA replication, and so there's two, actually, sorry, no, it does not undergo DNA replication. Rather, one strand of this double-stranded DNA is nicked, and then a single strand can be sent across this pilus into the new cell. As soon as that happens, DNA replication occurs, and so you create new strands on both of the Watson and Crick original strand. So once this enters into the new cell, DNA replication occurs. And so in the end, you have a full copy of two plasmids. So you have an F factor from here and an F factor now plasmid in here. Ligase closes, so you have a linear piece of DNA, and then the ligase closes this gap. So ligase is an enzyme that's actually able to covalently bond DNA strands, ends of DNA strands together. So this, when we talk about genetic exchange in this way, what we're talking about is that there's a DNA plasmid that was originally not in this other cell, but we've exchanged it and added to this other plasmid. So what is important is that this F factor contains genes that are necessary for this plasmid to function. So it has genes that are necessary for creating the pili. It also has genes that are necessary for synthesizing the plasmid, but it can also have additional genes on it as well. And so this becomes important for things like antibiotic resistance, because it might contain a gene that confers resistance to the antibiotic streptomycin. And so this is why it's so bad for humans, is that if a resistant gene evolves in a bacterial species to streptomycin, it's actually able to transfer that resistance to a large number of other cells. And this breaks down species barriers, where it's actually able to transfer it to all sorts of different species. So you might have an E. coli bacteria that's living in your gut, and if you do a lot of antibiotics, it might end up, evolving resistance to this antibiotic, but then it's able to actually transfer that, to pathogenic cells that can then evolve, that can then become antibiotic resistance because of this whole mechanism. So this plasmid, this plasmid mechanism is actually very, very problematic from a health perspective. Okay, so I mentioned before that there's kind of two different ways that conjugation can occur. And so this first way is when you have a plasmid that is transferred to a new cell. But you also are able to undergo this very different type of DNA transfer. And this occurs because the F factor is actually able to integrate itself into the bacterial chromosome. And so when this happens, we can see this because we have the orange original bacteria chromosome, and then in between that A and E gene, we now see this plasmid piece of DNA. So this plasmid is still able to create pili, but you lost this plasmid within the cell. And so when this happens, this F plus cell is converted to what's called an Hfr cell, high frequency of recombination cell. So again, this Hfr cell is able to conjugate with an F minus cell. So it's able to create this pili bridge, and it's able to transfer DNA from one cell to another. But now you'll notice the plasmid DNA is actually integrated into the chromosome. And so you start again from the same position within the bacterial plasmid, so you still have this nicking that occurs, but now you're not just sending over plasmid DNA, but you're also sending over some parts of the chromosome as well. So this is sent over, and then you have a certain amount of the chromosome that gets transferred. So because the chromosome is so long, it's 4 million base pairs long, you don't transfer the whole thing. You only transfer part of it before kind of a breakage occurs. DNA replication again occurs, so you end up having a linear strand now of DNA, and this linear strand of DNA is not actually ligated together. This chromosome, you have DNA replication, and so you end up having the Hfr cell is pretty much unchanged, so it's still in its original state. And so now what you have, though, is a recipient cell that is F minus. It has a linear piece of the chromosome from the other cell, and this, to designate the difference in alleles, we indicate this big A, big B versus little a, little b. But you can imagine there's homologous regions of the chromosomes that can kind of line up. So when this happens, this F plus cell, this F minus cell does not become F plus, because the parts of the plasmid that are necessary for building the pili aren't actually transferred into the cell. So you only have a small part of the plasmid, this little blue right here, that's transferred. But what you can have happen is what's called recombination. And so recombination can occur where this chromosome gets replaced by the linear piece of DNA. And so you end up creating this chimeric version of the chromosome that has the alleles from the donor cell integrated into its chromosome, and then it basically loses this B and A right here. So again, you have this Hfr fragment, so I'm showing you the location of 3 genes, and the assumption is that these 3 genes have different alleles in the recipient. So on the recipient chromosome, you have Lu minus, Arg minus, and Met minus. And then on this Hfr fragment of linear DNA, you have Lu plus, Arg plus, Met plus. And so you have to have 2 different regions that recombine. But when that happens, you can replace part of the recipient chromosome with the other. And so you just kind of like follow this crossover and come back. So this cell right here would be converted to a Lu plus, Arg minus, Met minus. If the recombination points occurred here, then this recipient cell would be converted to Lu plus, Arg plus, Met minus, or right here. You can also have multiple crossovers occur, where you have 4 total crossovers, and you end up having, Lu plus, Arg minus, Met plus. So you can have this conversion as well. So what are some differences between Hfr and F plus? So the Hfr strain does not make the recipient cell become also F plus. So in the Hfr strain does not convert the cell to become fertile. And the other main thing is, so when you have an F plus, the recipient becomes F plus, but with the Hfr to the F minus, the recipient remains F minus. Okay, so experimenters have spent time using this process in order to map the order of genes on bacterial chromosomes. So this was very important early on to demonstrate, for example, that the E. coli genome was circular, and it was also very important for determining the relative order of genes on the chromosome. And so the idea of this was to create this interrupted mating technique where you allowed Hfr and F minus strains to be incubated together so they could begin conjugation, but you interrupted the conjugation at certain times. And the way you did this is you actually just like put them in a blender, and then the shearing forces from the blender would actually break apart the pili, and the cells would separate from each other. So in this way, you could actually transfer a certain amount of DNA up to a certain point. And so the idea behind that is that you have this Hfr strain. This Hfr strain has the plasmid right here, and you allow it to start conjugation. So the DNA that gets transferred, oops, sorry, I think I drew this incorrectly. Sorry, this was correct, sorry. All right, so this right here is the origin, and then this is the sorry. All right, so this is the origin right here. So the Hfr strain starts from the origin of transfer, which is on the plasmid DNA, and then it begins to transfer this DNA kind of in this order. And so you see the origin is the first thing that makes it, and then you have the orange, and then the light purple, and then this other color orange, and then this green get transferred kind of in order. It actually takes a while for this to happen, though. It takes multiple minutes, on the order of an hour to transfer this DNA to the cell. So the idea was that if you interrupted this after 10 minutes, so you allowed conjugation to occur for 10 minutes, and then you interrupted it, at that point you would only just starting to be reached this first gene right here. And so if you actually looked at how many conversions occurred, so conversions where this allele exchanged with this allele, you would actually see a very small amount. Then after 15 minutes you'd see this huge jump because you'd kind of reach this point right here, and then after 20 minutes, you would reach this point right here, or kind of both of them are. And then after 30 minutes, you would be reaching here. And then after 50 minutes, you'd be reaching this gene right here. And so this is how you can order it, is it because, one, it takes more time to start to see these conversions occur. So you'll notice the Hfr strain and the F- strain have opposite genotypes at these four, and so this allows you to determine when a conversion has occurred. And so for an incursion to occur, AZI resistance, so this would become AZI resistance, and so you can see after 10 minutes it starts to happen, and then eventually about 90% of the cells would have that happen. And then the TON is replaced, it starts to be replaced after 15 minutes, and then approximately 80% of the cells reach that. So one thing you'll notice is the delay in time, but then you'll also notice that the maximum amount of cells that can be converted goes down from 90 to 80 to 40 to 25%. And so the reason of that is that this process is not 100% efficient. So the longer piece of DNA that gets transferred, eventually some cells will just have breakage occur or shearing occur that's unrelated to the blending, and so that's kind of why this percentage drops over time. So you should be able to interpret these graphs in order to figure out what your mapping is, the location of the genes, and you're mapping them relative to where this plasmid has integrated into the chromosome. So this is called time mapping. And so if you have different Hfr strains, so this is an example of where you have 4 different Hfr strains, these Hfr strains will all integrate in different locations on the chromosome. And so based upon this location, so it's also important to note the plasmid can be integrated in either direction, and so the direction that it starts can be either clockwise or counterclockwise. So based upon that, you can have this kind of random difference. And so when you did the mapping of this strain right here, this was the order that you got. When you did the mapping of this is the order that you got, and that's all relative to the positioning of the origin of transfer. All right, so finally, we will go in more detail about this Hfr strain, and kind of the activity will cover this a little bit in more detail. But one final thing I want to cover about conjugation that the book covers is this F prime. So an F prime occurs when an HFR reverts back to the F plus state. So we have the X factor, and it is integrated into the genome. And then what you can have is an excision of the F factor where it goes from the chromosome back to the plasmid. So the mechanism of this we won't cover in detail, but you can hopefully imagine how that occurs. This excision is not always perfect, and so you end up pulling more of the DNA out than just the original F factor DNA. And so now what you end up having is this plasmid that has the two of the genes from the chromosome that is on the plasmid. And so again, this F prime cell can conjugate and transfer the plasmid to a new cell. And so if this happens, what you end up having is what's called a merozygote. And so this merozygote is a diploid, it's a partial diploid, and it's partially diploid at whatever genes the bacteria plasmid has. So you'll notice that you have the big A and the big E from the host cell, and then you have the little b, little a from the recipient cell. So in this way, you're actually able to create like a partial diploid bacterial cell for some of the genes. So the F factor is an example of a plasmid, which we've already covered. And finally, I just want to cover transformation. So transformation is a different, so now forget about conjugation, forget about the F factor, forget about the pili. But the idea is that you have a bacterial cell. This bacterial cell might lyse, and so what you end up having is pieces of DNA that come from this bacterial cell. And so this bacterial DNA, the bacterial cell, the recipient cell, is actually able to import these pieces of DNA into the cell. So the DNA enters the cell, it separates the double-stranded DNA into single-stranded DNA. One of them is degraded, and then the other one, again, can be used as a template for recombination. So if there's homology between the chromosome and this linear piece of DNA, then you can end up exchanging the DNA, which is shown here, to create this chimeric. So kind of the process is very similar with the HFR, where you transported a linear piece of DNA into the cell. The difference between transformation and conjugation is where that DNA comes from. So for transformation, it's just environmental DNA that's taken up. So you have to both import the linear DNA, and then you also need to have some sort of recombination in order to have a permanent change in the recipient cell's genotype, which requires homology. And then co-transformation just indicates is analysis of how frequent two genes. So you have this piece of DNA that you have, and then code transformation is just when you have multiple genes that are exchanged. All right, so this is the extent of what we will cover on conjugation and transformation in the lecture.